The field of genetics is traditionally broken down into three major areas of focus. The first is transmission genetics, and this is probably what most of you think of when you think about genetics. This is the section where we learn about heredity and how traits are passed on from one generation to the next. So the focus here is on the individual, what the effects of that gene are, genotype, phenotype, maybe where they're located on a chromosome, how their inheritance of different traits comes from their parents, how those traits or alleles interact between those parents. So in this section, we're going to learn about cell division, we're going to learn about Mendelian genetics, and then we're going to learn about some of the more complicated patterns that we see in inheritance. Our second division of genetics is molecular genetics. This is my favorite part. It's not everybody's favorite part, but it's my favorite part. In this section, we learn about the chemistry of the gene, so the physical structure of DNA, or I'm sorry, I should say the chemical structure of DNA, how all of the different functional groups within the molecule affect the behavior of that molecule and how it can actually be used as a coding molecule. Part of that is also how it can be used to express our traits. So in this section, we're going to talk about physical structure of chromosomes. We're going to talk about gene regulation. We're going to talk about the processes of replication and gene expression, how we go from a gene or a genotype to a phenotype. So I would say the bulk of our class actually falls into molecular genetics. The first unit is mostly transmission. Units 2 and 3, and even into 4 a little bit, are really more molecular genetics. Now, don't let molecular scare you. We will do a little bit of chemistry, but it'll be on the level you learned in 101 as far as physical structure, chemical structure of the gene, and we'll talk about some of the properties there and then how they affect all of these other processes and the overall structure and function of chromosomes. The third division of genetics is population genetics. You could take a whole class on molecular genetics. You could take a whole class on population genetics. We spend the smallest amount of time on population genetics here, which is a little bit unfortunate. Our textbooks only spends a little bit of time on it, and really because this is where genetics overlaps with evolution and ecology. And so we start to bring in other fields of biology here, and very quickly we can get into tons and tons and tons of statistics and population dynamics, and then move very quickly into ecology. So we only do a little bit of this. This is another of my favorite parts. If you haven't noticed yet, I just love this class so much. But this is really, I think, where genetics shines as far as why it's important. Obviously, it's important, genotype, phenotype, medicine, all of that, but population genetics is really where we get to the heart of why it matters for biology. The other sections are going to tell us why it matters for us, why it matters for medicine, why it matters for agriculture, but population genetics is really where we learn why it matters for biology. All right, so humans have been using genetic techniques for years and years and years. About 10,000 or so years ago, humans started domesticating plants and animals, and this is where we really began using genetic techniques and artificial selection to cause our domesticated plants and animals to exhibit the traits that we wanted. The understanding of genetics is also present in ancient writings. Jewish people had an understanding of the genetics of hemophilia. They were studying parent and offspring and expression of those genes. They probably didn't understand a mechanism, but they did understand the pattern and how it was actually being passed from parents to offspring. and then the ancient Greeks, and then everybody after that was really looking at how traits are inherited. And we're going to talk about some of the different ideas, the different foundational ideas for how traits are actually passed on. Some of them have been supported by further biological study and evidence, others have not. But they're all of those theories of inheritance are important in shaping how we see genetics today. So very, very early understanding of genetics. And keep in mind that most of what we're going to talk about today came from an understanding without any vehicle or mechanism for passing on information. People knew that offspring looked like their parents. They knew that parents passed specific traits. They knew that individuals were a combination of those traits. They could see that. You all can see that when you look at your biological siblings, biological parents. But in the absence of any vehicle or mechanism, they had to get pretty inventive. And we didn't have a vehicle until early 1900s when we start to see the chromosomal theory of inheritance, which basically says that our genetic information is contained in chromosomes. And then it was another 50 years until Watson and Crick, Rosalind Franklin, and Maurice Wilkins were looking at the actual structure of DNA. And they were not the first, but they were the ones that sort of got us there. It wasn't until the 1950s that we actually had a structure and a more concrete mechanism. So for most of human history, there was no understanding of the vehicle or the mechanism. So early people subscribed or believed in something called pangenesis. Now, pangenesis says that each part of the body holds its genetic information for that particular part. So, for example, your head contains the genetic information to make a head, and that information travels to the reproductive organs where it is put into a little particle called a gemmule, that the particle that carries the information is called the gemmule, and that gemmule provides information to the embryo at conception. So let's say information comes from the brain, it's passed via a gemmule to the reproductive organs, either the sperm or the egg in the sperm in the male, the egg in the female, and then that information through the sperm and the egg is passed on to the zygote. Now, if you think about a lot of these, you can probably figure out how they make sense, why they came up from. Some of these are going to sound a little crazy because we have so much more information now and a better understanding of the mechanism. But for most of these, you can kind of think about and understand where they were coming from. It probably makes sense that the brain or the head codes for information for the head because that's where you see the similar characteristics. For a very long time, people recognized that there was a transfer of characteristics from parents to offspring. Now, how that transfer actually worked wasn't familiar, and the nature of those characteristics, or sort of the background for where those characteristics come from, was a lot of debate for a long time. Now, keep in mind that there's a lot of evolution that explains this. Prior to Charles Darwin, who we'll talk about in a minute, and even still today, there is always that overlap with evolution and religion and the idea of creation and a creator. And so a lot of this, especially as we get closer to Charles Darwin, a lot of this was new information. So in the 1700s, a man named Jean-Baptiste Lamarck, who was actually really important for biology, is kind of remembered for getting it wrong as far as inheritance of acquired characteristics. But Lamarck gives us our classification system for living organisms, our naming system for living organisms. He was really, really interested in all of the diversity on Earth, and he was trying to explain it. He was trying to categorize it, and he was trying to explain it. So his explanation for the variety of characteristics that we see in living organisms is that traits are acquired during a person's lifetime. And then during their lifetime, that information is sort of deposited or recorded into your hereditary information, and then that information is passed on to the offspring. So he got a little bit of it right. That information is definitely passed on to the offspring. His mechanism has not been supported by later biological evidence. In general, a trait that you acquire over your lifetime is not passed to your offspring. So the example that I like to use here is if you are a bodybuilder and you bulk up your muscles and then you make a baby, that baby's not born with those bulky muscles. They have to then acquire those muscles over their lifetime. So the sort of classic example of this, besides my favorite, the bodybuilder one, is giraffes. And so the idea here is that if you imagine a giraffe that's kind of shaped like a zebra, and when it's born and then over its lifetime, this first giraffe stretched out its neck, and then it passed that stretch onto the next generation, and then that generation stretched its neck a little bit more through their lifetime and passed on that neck. And if you think about it, you might be like, well, yeah, that kind of sounds familiar. It's not exactly the theory of evolution that Darwin gives us that is supported by a lot of evidence. So remember, this is traits that are acquired during the lifetime. So what this idea is saying is that based on what you do during your life, you can change your physical characteristics and thereby change your genetics. And that's where we get the problem here. Basically, genetics is, I like to say, you get what you get and you don't pitch a fit. You're born with whatever genetics you're going to have. Those genetics don't change throughout your lifetime. Your genes are your genes are your genes. You get what you're born with, and they remain your genes. Evolution is actually acting on the genes of the entire population, not the genes of an individual. So one of the crazier ideas, at least I think it's crazy, is the idea of pre-formation. And again, you can probably imagine how people came up with this. Basically, the idea of pre-formation is that inside the sperm or the egg, there is a fully formed miniature adult. So what this would mean is that in either the sperm or the egg, the little tiny individual is already there. So what this is saying is that the individual does not come from a fertilized sperm and egg that has then divided, which is what we know now. Basically, that they're already contained in there, and basically just enlarge until they are a baby, until they are born. This little structure is called a homunculus, which is kind of funny. There's a little diagram here on the right showing the homunculus with a giant head that I don't quite understand. But if you think about this one, it sort of makes sense. A baby is born formed, and individuals, doctors and whatnot, probably saw premature babies. They probably saw animals who had little teeny tiny babies, and they probably weren't really seeing all of the molecular structures. So they definitely weren't seeing an immediately fertilized egg. They weren't seeing the initial formation of cells. They were seeing this small human that is born, and so they just worked backwards and said, well, that small human must already be present in the sperm or the egg. Right, so we're getting kind of closer to Charles Darwin and then closer to current time. As far as explaining where the traits in individuals come from. People knew they were passed from their parents because you could see that. The general idea was something called blending inheritance, which basically means that offspring are a blend or a mix of parental traits. Now, what I don't mean here is that you have your mom's eyes and your dad's nose. What I mean is that if we look at any particular trait, say the nose, that you are a blend of both of your parents' noses. Some of you are, based on your genetics. But if you take all of your characteristics together, you are not a blend for all of your characteristics. Most of you have a distinct characteristic that is similar to one parent or the other. So the idea of blending is that you kind of take the two extremes of your parents and the offspring get an intermediate. So our example here are garden peas. We'll see those again when we get to Mendel. And so we see this example here. We have a smooth pea and a wrinkly pea. These are normal pea phenotypes. And so blending would tell us that the offspring would be semi-wrinkly. Another way to think of this is if you have an individual with straight hair, and has a child with someone with curly hair, their hair would be wavy. That's kind of blending. We do see something similar to blending, and please don't get this mixed up. You might even be thinking about it right now. Not codominance, incomplete dominance. Incomplete dominance does appear to have a blending type appearance, and we'll get to that a little bit later. But definitely not all traits exhibit incomplete dominance, and most of our traits are not showing blending. Most of our traits where we do see sort of a range, so if you have a tall parent and a short parent and you're right in between, that's actually many genes working together. So there is an explanation for when we do see some of these blending type traits. So this is just a really brief timeline of some of the different stages of genetics. I'm going to just let you pause and look at this if you're interested. I'll have this in class also. I'll probably post it on Brightspace. As we go through the semester, you will be asked to know some of these dates. Right now, for Chapter 1, you don't need to know these dates. All right, so then we get to the mid-1800s, 1850s or so, and we get to Gregor Mendel. You probably are familiar with Gregor Mendel. He's considered the sort of grandfather of genetics. Gregor Mendel discovered the basic principles of heredity and sort of set the foundation for our understanding of heredity. He was working in a garden with pea plants and just following thousands and thousands of pea plants and looking at what happens when you cross two different alleles, two different phenotypes for the same gene. And he kind of gave us a basic understanding of dominance and recessive, and the idea that each parent contributes a little bit to the phenotype. For Gregor Mendel, he didn't have a mechanism because this is not necessarily before all microscopes, but before wide microscope use, and definitely before the use of good microscopes, and definitely before the time of molecular biology. So he didn't have a mechanism, which caused some problems, but he did give us just sort of this general broad understanding of the basics. And that was going to be really important when Charles Darwin comes on the scene, and then later when we get the molecular nature of the gene. In the sort of all around like late 1700s, early 1800s, mid 1800s, we had several people who in different writings and whatnot put forth what we call the cell theory. So the basic idea of the cell theory is that all life is composed of cells. All cells arise from pre-existing cells, which there we have some reproduction, and we also have some information transfer. we have to have a mechanism of transfer of information. If we're saying that one cell comes from another, we also have to assume that the information for life comes from the previous cell. And then the cell is the fundamental unit of structure and function, meaning that a single cell can perform all the functions that we consider living. I should say a cell does all this. So the other way to say this is the cell is the smallest unit of life, the smallest one that we consider living. We're not going to go into the cell theory too much. It is important, but at this point, it's not too important. Just kind of keep this in mind. We're going to talk about reproduction in chapter 2, and then, of course, how we pass that information on a little bit later. All right. So really, until we got microscopes, there was no theory for a vehicle for the transmission of genetic information. Our first sort of widely published information on the structure of cells comes from Walther Fleming in 1879. He was observing cells under a microscope. I should say he's not the first one to observe cells, but he was working specifically with chromosomes. These are some original cell diagrams, early cell diagrams. These are obviously not photographs, these are drawings of what Walther Fleming and some others were looking at under a microscope. And it is from here, this early observation of chromosomes and chromosomes dividing during meiosis and being passed on to the next generation of cells. Mitosis. I got ahead of myself. I was thinking cell division, and we're going to focus on meiosis. Walther's diagrams of chromosomes really helped people understand that there was something being passed from one cell to the next, and it was those chromosomes. So that gave a potential vehicle for genetic information. And from there, people started really looking at chromosomes. One of the most famous was Thomas Hunt Morgan, and he worked with fruit flies, and he gives us a lot of information about fruit fly chromosomes and really supports the cell theory and also the idea of heredity with some of his work on fruit flies and chromosomes. Then we get to Charles Darwin. You should all be familiar with Charles Darwin. In 1859, he published his theory of evolution through natural selection and basically said, hey, organisms change over time. And his problem was that there was no mechanism. He didn't have a mechanism. This is right around the same time, maybe even a little before Fleming was working on chromosomes, and definitely before Thomas Morgan was working on flies and chromosomes, and really before the field of cytology, which is the study of cells. And so he didn't have a mechanism. He had all of this evidence that through the process of natural selections, populations change and traits in the population change, but he didn't have a mechanism for it. And so Darwin, and along with Alfred Russel Wallace and his support of the theory of evolution, between the two of them, they had this really great case for natural selection and change over time. But they were not able to propose a mechanism or a vehicle for these changes to happen. Then we get getting a little closer to early 1800s. Early 1900s, late 1800s. We get the germplasm theory. And we're starting to get to things that we know a little bit more. This theory says that cells in the reproductive organs, so in other words, sperm and eggs, carry a complete set of genetic information. So this is a little bit different from pangenesis, which says that each of the cells contribute a little bit. Pangenesis is not saying that the sperm and egg don't have all that information, but what it is saying is that the sperm and the egg have always had all that information. It doesn't come from somewhere else like we see in pangenesis. The sperm and the egg have all of the information already, and that is passed on to the offspring or the zygote. Zygote just means fertilized egg. So I should say fertilized. The zygote is a fertilized egg. So the sperm and the egg pass on all of the information, which means the zygote has all of the information. And from that, what this means is that all cells have all of the genetic information. So each of our cells in our body is going to contain the entire set of instructions. Early 1900s, we get a couple of really important bits of information, and I think I might have written the wrong date for Thomas Hunt Morgan. Sorry, a little bit earlier. In the early 1900s, we have Walter Sutton and Theodore Boveri working on chromosomes, and they both identified genes as being located on chromosomes. So this is called the chromosomal theory of inheritance, and we'll get to this a little bit later. We'll cover it in more detail, but basically this is where we get that vehicle for passing genes from one generation to the next. Sutton and Boveri said genes are located on chromosomes. Our specific instructions are located on chromosomes, and those chromosomes are passed to offspring. And then shortly after that, in 1910, Thomas Hunt Morgan, who is studying fruit flies, notices that genes are located on specific chromosomes. So genes have a specific location on a chromosome. And Thomas Hunt Morgan is famous for his work with fruit flies and eye color. So what Thomas Hunt Morgan noticed is that eye color, specifically a white-eyed mutant or white-eyed mutation, is located on one of the sex chromosomes. And again, we'll talk about this experiment later, but this was important because Sutton and Boveri said our genes are located on chromosomes, and then Thomas Hunt Morgan supported it and said, yes, and here's why. Here's where one specific one is located. It is located on a specific chromosome. So he was able to support their theory. Finally, we get to 1953. This is where we start to see modern genetics. Depending on the age of your parents, they may or may not have been born. My parents were actually born after this. Both of my parents were born in later 1950s, but definitely in the time frame of my grandparents. So essentially within my grandparents' lifetime is when the entire field of modern genetics has come about. So in 1953, James Watson and Francis Crick published a paper showing the structure of DNA. This was supported by work by Rosalind Franklin and Maurice Wilkins, a little bit shady to be perfectly honest, but we'll talk about that a little bit later. And once we get the structure for DNA, we get a mechanism or a proposed mechanism for replication and gene expression. Now, the actual mechanism will come later. All of the parts and components will come later. But once Watson and Crick published this structure, they immediately were able to look at it and say, we can now see how DNA could be replicated and how DNA could be expressed. And this is where we really start with what we would call the modern field of genetics. And this is sort of in conjunction with what we know from Gregor Mendel. And then later we'll see how this relates to Charles Darwin and evolution. Gregor Mendel didn't have a mechanism. Now with the work of Sutton, Boveri, Morgan, Fleming, we have chromosomes, and now we have a structure for those chromosomes.